All right. Well, good morning again, everyone, and welcome to day 2, session 4 of the covidClassifyR Shiny App workshop. Today, we're going to go over the data preparation and how to go from exporting your data from the Luminex machine to importing it into the Shiny app. So just to recap, because it was yesterday, I'm not going to go over, what we went through, but I've just popped all of the links to the different session materials there for you if you do want to revisit these slides later. And just a reminder that for session 2, we will be sharing those confidentially because of unpublished data that is in the presentation. And of course, you can always go to the workshop website if you want to find the materials. So as I mentioned before, we're going to go over how to prepare your raw data file and your plate layout. I'm going to kind of sprinkle throughout the presentation some do's and don'ts of what you need to be doing so that the application can actually process your data. And we'll go through how to import the raw data into the application on the web. And there you can find the application website. Some of you may have already visited it, I hope, and maybe explored a little bit. All right, so preparing your data. Whoops, sorry. The raw data requirements for the application, as we mentioned briefly yesterday, your raw data should be exported from the Luminex machine in Excel format. You want to make sure that your sample labels in the protocol are as follows. And so we touched on this yesterday. Fiona also went through this. Your standards should be labeled with an S and then a number as required. So you have your standard 1 or S1, S2, S3 sequentially as needed. Your blanks should be labeled with a B. And then if you have more than one blank, you will want to put blank 1, blank 2, etc. And then on the Luminex machine, as Fiona described yesterday, you don't want to include individual sample labels or anything. You just leave the samples as unknown. And then just reminding you that, take the time to kind of plan out the experiments that you're going to be doing. If you're going to be running a bunch of samples from a cohort or a cross-sectional survey, make sure that you set up your protocol for the running of all of those plates and all of those samples, and keep consistency with your antigen labeling. And this will make it easier for you down the line when you want to merge all of your data and you want to analyze it all together. And so it's a good thing to just think about beforehand and then put into place before you start so that you maintain consistency throughout your project. And just so that you understand essentially what the Shiny app expects, it will interpret anything that starts with an S as a standard. So it will use the antibody data and the MFI readings from these samples to calculate your standard curve. So if for any reason you labeled your blank starting with an S, I don't know why you would do that, but maybe it's a typo, it will actually include the antibody data for that blank sample, and we don't want that to happen because then our standard curves will not be accurate. And then similarly, it will interpret anything that starts with a B as a blank. And so in the app, it'll show you what your blank samples look like, but it will ignore them for all of the calculations because it's not actually an unknown sample. And then anything else will be considered an unknown and will be analyzed, as we'll describe later. Now for the plate layout requirements, similar to the raw data, your plate layout should be in Excel format. And for each 96-well plate that you run on the Luminex machine, you will prepare a plate layout that is specific to the Shiny app. And this will include the sample labels that will match your raw data. So this is what the Shiny application will use. So you supply this, and then that's how the Shiny application will match your raw data values to the corresponding sample based on the plate layout that you have imported. So this is where you basically tell it, this is, well, A2 is actually my second standard, and then the Shiny app will match everything together. I'm gonna sound like super repetitive, but just really, it's super important to label your samples appropriately. And the only difference in the plate layout to the machine is that now this is where we actually label our unknown samples according to the specific sample codes. So for example, this ABC, let's just say your cohort study or your cross-sectional survey, the study code is ABC, and then the numbers are for individual samples. So you can then label them, ABC 001, ABC 002, instead of unknowns. And that's how this Shiny app will match everything together. Another thing to make sure is that there are no extra cells filled out other than the plate wells and sample names. And so the Shiny app is designed to expect just a 96 well plate table, essentially. So if you have notes, below for yourself, that's fine for your own run, for your plate layout for the Luminex run. For example, if you want to keep note of, which antigens you used or which bead regions, that's fine. But then you need to create a separate file for the Shiny app, which is just your plate layout, because if not, it won't work. My suggestion would be to just use the example plate layout as a template and then just fill in with your sample details each time, because that one is prepared as it should be. The other point I wanted just to just make today, if you haven't run a full plate, which sometimes will happen, maybe you're just running a repeat of some samples that didn't work, you might not have a full 96 well plate. That's fine. You just leave the wells with no samples completely blank on your plate layout. And similarly, on your machine, you would have only selected unknown samples where there are samples. And so it will all match accordingly, and it will run. The other point that Fiona also brought up yesterday is that you want to make sure that the word plate is included in the first column. And this is important because the Shiny app expects this word so that it knows where the relative positions are of the 96 well plate. So it uses kind of that as an anchor to say, okay, this is the top left corner of the plate, and then it can kind of it can figure out which location your samples are in, and then it matches them accordingly. So I've popped the link there for the example data. Some of you may have already downloaded it. If you haven't, please make sure to do so after this session. We've got a week until the next one. So do maybe spend 20 minutes just kind of looking, or maybe not even 20 minutes, maybe just 10 minutes going through the example data, making sure you kind of know what it looks like. And here is just a screenshot of what the example raw data Excel file should look like. And this is a direct export from the Luminex machine. So I'm not going to go into what the Luminex exports, because it's, there's a bunch of stuff that probably is not even useful, but it's just to familiarize yourself with what it should look like. And similarly for the plate layout, this is what it looks like in the example data. So here I'm just pointing out that plate word. This is where the Shiny app is going to say, okay, great. This is A1, and then directly below it is row A, directly to the right is column 1, and that's how it kind of reorients itself in the plate so that it can match the data for your samples. Here in the example, we've got 2 blanks, and so they're labeled 1 and 2, and then our standards are labeled with S and numerically as required, and then everything else is labeled with our sample code. So we have 84 unknown samples in this plate example. And like I said, you can just use this layout for yourself. All you just need to do is sort of clear the contents from the plate and then insert with your values. And that way you don't have any other cells from column N onwards or row 10 onwards. All right, so I thought we'd do just a little quiz. So please don't be shy. And it's so close to me telling you the answers, so I'm sure everybody already knows the answers. But anyways, just to refresh your memory, how do you need to label your standards? Anybody want to type in?
S1, S2, S3.
Whoops, sorry, couldn't hear you that well.
S and numbers.
SN numbers, excellent. So yes, you need to label them starting with S. How about, what would you do if you have more than one blank sample? Maybe someone else, because I know that was Daisy, wasn't it? Daisy's super familiar with this. So you're not allowed to answer. Sorry, Daisy.
I think for this one, blank with different, B with different numbers like B1, B2.
Excellent. Yes, so you would label them with sequential numbers. How do you need to label your unknown samples?
So the plate label here.
Yep. Yep, you can use your study codes. I should have their in your plate layout. So I might add that later. Yes, you can use the study codes in your plate layout so that you know which unknown sample is which. Excellent. So moving on to now actually importing the data into the app. Now, so based on the experience from the Molecular Hub, seems like this was quite easy. So very glad to hear. So now I'll take all of you through what that would entail when you do it on your own. So first of all, this is sort of the app. Once you're on the web, you want to firstly navigate to the import tab. So this is, if we were to click there, this is the page that will appear. And as I showed you briefly yesterday in the overview, you have a bit of a, some instructions there. Obviously, all of you will have gone through the tutorial many, many times. So this will be, this will be really just the same news to you. But in any case, you can also just kind of read here for some prompts. You can also download the example data again here if you need to, although you should have already downloaded this. And then we have this sort of experiment info panel where you have the experiment name and other details, which I'll go through later. And then here is sort of your sanity checks that everything's imported correctly. So for the experiment info, you can type directly in this box to change the experiment name. So what do I mean by experiment name? This is sort of, this will depend for everybody and depends on how you label your different plates for your projects. My suggestion and example would be my experiment name, plate 1. So for example, the ABC study, plate 1, the ABC study, plate 2, whatever is useful to you, and just, maintain consistency as you can throughout the study for you to then be able to collate all your data. You're also able to enter a date. And the way I envisioned this was, the analysis date, because it's the date when all of your process data files were generated. So, I think the default, which is today's date, is probably the best, but maybe you need to change that for whatever reason. You can, if you need to, you just click and then select the date on the calendar. Although my suggestion would be to keep the date as the analysis date. And then finally, there's a box here for any experiment notes. And this is really, you don't have to use this if you don't need to. I just envisioned, maybe sometimes something will go wrong and you just want to keep track of it. I accidentally, put the sample from B2 and B3. So disregard data for B3, whatever makes sense to you. Or maybe you want to indicate this is a rerun of plate 1, or anything. We're all lab people. We know that there's tons of different little notes to yourself that might be helpful, and sometimes you don't need them at all, but you do have the option there. And all of this experiment information will be displayed on your quality control report when you download it. And so then, when you do download this and you keep a record of each plate that you've run, you will also have these details for yourself to remember. If you go back to the data from 2 years ago, you'll remember, oh, right, that's the plate where I had to repeat 5 samples or whatever. And then you can see here, this is where you're going to upload your files, which I'll go through in a bit. Another tip that I have for you about experiment names and sample codes, it's probably a good idea to
Yeah, Bob just sent a text, a message.
Sorry?
Bob sent a question.
Yeah.
Yeah, can they run info from the MacPix automatically populate this tab? I think when you were talking about the experiment nodes, what can you confirm?
So if it can be pre-populated from where, sorry?
Hey, hi, Shazia.
Hi.
So my question was, instead of typing all this when we're importing, can it automatically
Be transferred from the
populate?
MacBooks.
Yeah, from the Mac, because when we set up the run, you'll type up all this stuff like experiment name and the date.
But the date, so the date that you run the plate may be different to the date that you analyze. If that
Okay.
makes sense. So I think in terms of the date, it's probably good to still, I mean, ideally we'll be running a plate and running the QC on the same day, but we all know that that's not always the reality. So I think keeping track of when you run the plate and when you process the data is probably a good idea. But in terms of the experiment name, yeah, that's not a bad idea. I guess I just wanted to make it more flexible in case, you know, someone needed to change that for whatever reason or, you know, but I think it would be possible. Do you think it would be too much extra work to have to put in those details in this section?
Oh no, it should be okay, but just, you know, in case we run too many plates and repeat too many things.
Yeah, no, I see. I totally understand what you're saying. I guess I was kind of envisioning having a bit more flexibility because let's just say what happens if you forget to put the experiment name in your Luminex run and then you can't, do you know what I mean? Then you can't edit it. So anyway, I can see, yeah, I know what you mean. So I guess the ideal scenario would be maybe not trying to not run or, you know, trying to avoid running 20 plates and then doing this 20 times, like maybe every 3 plates or maybe on a Friday, you do this for all of the plates that you've run, if that makes sense.
Yep.
But
Okay.
yeah, I'll keep that in mind because maybe it is something that we can just display on the QC report anyways. You know what I mean? Like it'll still import the information and then whatever you enter, it will also be included in the report. So I think that's a good idea. I'll see if I can maybe just have it always print out in the QC report automatically.
Okay, thank you.
No, thanks. Who was that? Sorry, because I can't see your face.
It's Sarah and Valin Goroka.
There. Okay, sorry, I still don't recognize your voices. Thanks for the question. And yeah, please keep interrupting me. Discussion is great. So yes, and just to finish off this point, what I was just suggesting to all of you is to avoid spaces and special characters in your file names and your experiment names. And in the case of this app, the experiment name that you enter here and the date will actually be concatenated for all of your process data files. So in this particular example here, I don't know if you can read this sentence at the bottom, all of your downloadable files, so your process data, your quality control report, etc., will be labeled as experiment 1 _ the date that you choose. So if we change this to Shazia's experiment, it'll be Shazia's experiment _ date. And so then that's how you can keep track of everything. So that's why I think the analysis date is important, because it's the day the data was processed. So when you download the process data, it has the date that it was actually processed and it will match the quality control report from that date. And so I think it's a good way to kind of keep track of what's happening, especially when we get in, you know, when we get on a roll and we start analyzing plate after plate after plate, and when we have, you know, thousands and thousands of samples, that hopefully this will help keep track of different things. So
Hey Shazia, so then I was just thinking, like, if we had a separate tab for maybe experiment date, and that.
Oh yeah, that's a good idea.
Yeah.
I should.
Analysis date should be the date of the analysis.
Yeah, let me make a note of that. That's a good idea. Experiment date or plate run. Yeah, excellent. I'll definitely incorporate that. I think that's a good idea. Great. So I just had a quick demo for those of you who haven't really entered into the app. You can kind of see how it works. So basically, I'm going to narrate what's happening, but it's literally everything we just went over. So you can type in the box and change the experiment name. So here I've just put, tutorial example with an underscore, no spaces. Change the date to the 1st. Again, I would encourage you to avoid maybe changing the dates, and then just in a note, this is a demonstration. And then that's it. So hopefully that should be quite simple. And I like the idea of including another box that is, enter the plate run date, for example, and then you can kind of keep track of when all of that is happening. I think that's great. Great suggestion. Thanks, Maria. Any questions at this stage or comments? Otherwise, the next step. Oh, yes.
The time for the experiment is automatically captured on the report after the.
Sorry, I can't hear that well. It's a bit far.
The time of the experiment.
Oh, yeah. Date and time. Yep, good idea in case you run sort of more than one in a day. Excellent. Thanks for your suggestions. And then the next step is just to upload your files. So really, it's really simple. You just click the browse button to navigate to wherever the files are in your computer. And then again, I would suggest you to try and, I'm sure all of you do this already. We're all, lab people. We've been doing this for a while, but it would be a good idea to have a folder with all of the runs, for all of the plates that are from a particular study. So the ABC study, I have a folder, then I have plate 1, plate 2, plate 3, and all of the files, the raw files and the processed files. So that's kind of how I would envision this can help you do that. But, obviously everybody has different systems, but that would be a suggestion from my end. And you just you click the browse button, you find your files on your computer, and then the bar should look blue and complete when the upload is successful. If you get an error, the first thing I would say is just make sure you're uploading Excel files and make sure you're uploading the correct file. Similarly, you would do the same thing for your plate layout. And here I'm just going to demo the uploading, and you'll kind of see how the app starts to react. So I'm just browsing for my raw data file, which is, I'm just using the example data here, and you can see the progress bar, and it's already starting to populate. So the app's already recognizing the files that you've loaded. So in the next section, we'll talk about these 3 tabs. So the first one was the check the raw data. So you notice that as soon as you uploaded the file and it uploaded successfully, it started to populate here. Now, I don't think this tab is overly helpful, to be honest, because it's just everything, but I think it's a good sanity check to just make sure, oh, okay, great. Yep. Oh, sorry. Oh, okay, great. It looks as expected. Yes, it was this is the protocol name. Yep. Okay, great. It's just like a quick check for you, but hopefully it can help you just make sure that everything is going all right. Because I know that the one thing that I thought about a lot when developing this is that sometimes when things are so easy, you can also lose track of what you're doing. So like, what if I uploaded the raw data for plate 1, but the plate layout for plate 2, that could also be, that could be a huge error that I wouldn't catch otherwise. So what I've done is on this top, you can really quickly check the raw data file name. So this should match what you're analyzing. So if I'm analyzing plate 2 and this says raw data plate 1, then I go, oh, whoops, wrong one. And then I'll go back and upload the correct one. So these are just little things that you can kind of check as you go to make sure that you are analyzing your data appropriately. The next tab is this check run info. And so this is basically from the same file, but it's just the run info portion. So I think this is what Sarah was talking about before, that you do kind of already have this information here. So I think I can build, I can build it so that it can go into the quality control report and some of this info as well. And again, here, I think people will be looking for different things. I always just kind of check the protocol name because I use the same protocol for all of the plates, which hopefully you will also start doing or are already doing. Sample volume, for example, the batch, which is the plate, and just little things like that. It's just another way to just quickly look at your data and make sure it all looks okay. And then finally, the check the plate layout. This is where, again, it's really important that your samples are labeled correctly so that they are interpreted correctly by the application. Here you can, similar to the raw data, you can make sure that the plate layout is the right one. Now with the plate layout, it's a bit easier, right? Because if you know that you're running samples 1 to 84 and suddenly plate 2, in plate 2, you would expect 85 to whatever, and suddenly it looks different, then you'd be like, okay, this is the wrong data. With the raw data, it's a bit trickier. So I've just included this for all of them. And again, make sure that there are no extra cells. So if suddenly I see note, or extra columns, then I know that I need to go back and fix my Excel file because the application won't actually process the data correctly. Any questions at this stage? All right. So finally, just to finish off this section, I wanted to just show you a demo of everything. So I know I've shown you little bits, but now we're going to go kind of from start to uploading data and checking the plate layout. So I'll narrate again as we go. See if I can do this. Yeah, so we go to import data, change the experiment name. Now it's called My Amazing Experiment. The date, I'm just changing it just to show you that you can. And then the experiment nodes, this is a demo. I'm uploading the raw data file from my local computer, and then I can upload. You can already check the raw data there. It's populated. As I said, it's not overly useful because it's literally the entire thing. The run info is just the run info, so you can look at this one as well, make sure it looks as expected. And then the plate layout uploaded successfully. So now I can check the plate layout and it looks great. So now we're ready for the next section, which is how to actually perform quality control of your data. And I've included just the acknowledgements and everything in all of the slides because they are standalone online. So I just wanted to make sure in case someone isn't following along all of the slides, but thank you to all of you again for listening and everyone who's supported this. Contact details again, and I've included all of the resources at the end as always. And I'll open it up to all of you for questions or comments, if there are any. Sorry, I just saw that, by the way.
I think this is close enough.
Yes.
Everybody's feeling all right with all of that?
Yeah, Shedrick has a question, but he's trying to form it. Yeah, articulate.
Yeah, no, take your time while you kind of think about it. I'm just going to switch over to the session 5 slides, but no rush at all. Just shout when you're ready.
Like if you're trying to optimize an experiment, and maybe you'll just edit as unknown and then make some notes underneath, or like if you're trying to test some different conditions for the particular.
Can you get a bit closer to the mic?
I don't know if it's a good question, but if you're trying to do an experiment and optimize some samples and make some conditions or remove some conditions from the from a particular well, how can you are you gonna put it as are you gonna leave the well as unknown and make some notes at the bottom, or?
Yeah, actually, it's a great question. There are no bad questions. It's a great question because the way I have done it in the past and have suggested the lab team here in Melbourne do it. So let's just say, and this will happen a lot. We're never lab work is never perfect, right? Of course, it would be amazing to always just run a 96 well plate and everything works perfectly and you're done. That's not how it works. Sometimes you might have to repeat. You might have some, 10 wells that are empty, so you use them to repeat some samples, for example. Or maybe you want to, like you said, try some different conditions. So what happens, if half of the plate is condition 1 and half of the plate is condition 2? How would, I'll throw it back to all of you. How would any of you do that in the application? And then, I have a suggestion myself as well, but I'll open it to anybody else if anybody has any ideas.
Yeah.
So maybe I'll jump in. Let's just use that example of having half of a plate condition 1 and half of a plate condition 2. The way I would suggest you can approach it using this app is everything that controls how the app interprets your data is the plate layout. So what I would suggest doing is having 2 plate layouts, even though it's actually the same raw data and technically the same Luminex run. It's kind of like 2 separate runs. So the way I would suggest to do it is to do a plate layout for the first half and just ignore the rest and process that. And then generate your processing and your QC report for that one. You would want to put in the notes, this is condition 1 from plate 2, for example. And then you rerun the app processing for the other half of the plate. And then you would write in your notes, this is second half, plate 2. And then you would fill in your plate layout only with the samples you want to analyze for that particular half. Does that make sense?
Yes.
Excellent. Great question. Any other questions from anyone else? If not, and if you think of anything, feel free to interrupt me at any time. There's no rush in thinking about your questions, or I know this is a lot of information at once. But it's great that you're starting to think about how, this might change in real life. Obviously, this is the example and the perfect scenario, but things aren't always that simple. Some other things that we've discussed here in Melbourne are different IgGs. So what if you were to run IgG and IgM on the same plate, for example? You would do the same approach. You would analyze the IgG together with the plate layout as the way to kind of tell the app only look at these. And then for the IgM, the same thing. So we have gone through some of those, I guess, not troubleshooting, but discussions here in Melbourne. And I do have a section in the tutorial that's troubleshooting. But I was thinking of making like a frequently asked questions section or something, and I'll pop these questions there, and just some suggested answers as they come. It was a bit difficult because we've been developing this for a while, but as we discuss them, I'll kind of pop them there so that you can always go back to that. And then the next session that we're going to go through is session 5.